All right, our next speaker is Andrew Stine. Andrew studies chemical and biological engineering at Northwestern University under Linda Broadbelt and completed practicum at Argonne National Laboratory.
Hello, everyone. Today I'm going to be talking about the known unknown, computational identification of promising enzymatic reactions and associated genes. So first, I need to start with what's an enzyme, because if you don't know what an enzyme is, this is going to be a really long talk for you. So most of the reactions that occur in biology, if they were unassisted, would occur much too slowly for life to exist. But fortunately, there are wonderful catalysts called enzymes, which can make these reactions happen much faster, in fact, fast enough for eating and living and all the other things we do to happen. And it makes cells happy because they make them alive. Okay, well that's good, but why are enzymes important? So this is my son Julian, and right after he was born, I vaguely remember through the haze of very little sleep that one of the first things that happened to him was that he had a blood test for phenylalanine hydroxylase. And it's because if you don't have this enzyme, if you don't have enough of this enzyme, and it's not treated, really bad things can happen. You can have developmental issues. It's very important that this is tested. But this is just one enzyme, and you have so many enzymes in your body, and we're just one organism. There's all these organisms out there, and a lot of these enzymes are very important for a lot of applications. So this is a picture of the enzyme. These applications include medicine, obviously, I just gave an example. Industrial biotechnology is essentially just using these enzymes. Biochemical catalysis and ecology. Unfortunately, it's very difficult to identify new enzymatic chemistry. It's because this is what, as a former Secretary of Defense once said, an unknown unknown. We still know so little about enzymatic chemistry that it's difficult to know if this given reaction, if it's not occurring because it's not possible, or just because we haven't found the enzyme that does that chemistry yet. So if you're trying to make some product P, you might be able to come up with a couple reactions that can get you there, and that's useful. But deep down, you're always wondering, are there more? Are there better ones that I don't know about just because we don't know enough chemistry? So we need some way to make predictions about enzymatic chemistry we have yet to observe. Of course, this seems like an impossible task. How are we supposed to predict something that we haven't seen yet? But fortunately, there's patterns in enzymatic chemistry. Certain types of reactions are quite common. For example, all three of these substrates are very different, but all three can do a very similar reaction. They can have this hydroxyl go to a carbonyl. So maybe we can just come up with a general rule that when you have a hydroxyl, you can turn it into a carbonyl. Well, this is great, but this is a computational talk, and we need some way to communicate this to the computers. So what we do is we represent molecules in matrix form, what's called a bond-electron matrix, where each atom has a row and a column, and the bond between the atoms is an entry in the column. So for example, this oxygen here is this column, and this double bond is this entry. This formulation also makes reactions easy to communicate to the computer, because all you do is you just make a matrix of how the bonds change across the reaction. And then to perform the reaction, you just need to add these matrices. So previously, my lab has implemented this scheme to create a program called BNICE, which stands for Biological Network Integrated Computational Explorer. But I really think deep down it just is someone wanted to spell out BNICE. How this program works is you load in a compound, and the program will look through the compound for any of these structures in the rules. And if it finds one, it applies the rule, and it generates a predicted reaction in a new compound. It then keeps going down the rules. It finds another one. It repeats this process. It goes through all the rules, creating all the reactions and compounds it can, and then you can load this new generation of compounds back in and create another generation. You can keep doing this until you create a network. And by exploring this network, you can often find novel pathways to compounds. So the best way to explain this is with an example. So I worked with an industrial collaborator, and they had a compound, which I can't tell you what it is, unfortunately, but a compound that's currently made from petroleum. However, it is a biological compound. Some organisms make it. Unfortunately, those organisms also make side products that are very difficult to separate from your product, making it not economically feasible. So they were wondering if there was some way that we could find pathways from pyruvate that avoid these side products and still make our product. So product is long to write, so I'm just going to represent it as money, because what they really wanted was for me to find a pathway that they can load in pyruvate and make money from this bacteria. So we did this. We looked for all the 4-step reactions we could find using BNICE to this compound from pyruvate. And we found several. Some of them actually went through these compounds we were trying to avoid, but many of them didn't. If you look closely, you'll see that all the reactions that don't go through this compound we want to avoid go through this single reaction step. All the pathways do. So we were wondering, is this just because we looked at rather short pathways, or is this going to be true for longer pathways? So we looked at 5-step pathways. We found that this was still true. Of the 1,400 pathways that are 5 steps, over 1,200 of them went through this step. So this reaction step seems to be the key to finding new ways to make this compound. So our collaborators went into the lab, and through a literature search, they identified 3 enzymes they thought could do this reaction. Because this is experimental work, 2 of them didn't work, but 1 of them did. And we found an enzyme that can do this chemistry, and it was not previously known to be able to do this chemistry. So we've identified new enzymatic chemistry. We're currently looking into whether we want to patent this, which is why I can't really disclose it. So I hope I've demonstrated that this BNICE, this rule-based procedure for predicting enzymatic chemistry, is powerful. But I actually sort of skimmed over how these rules have been made in the past. And how they've been made is basically graduate students have gone through databases of enzymatic chemistry, and then through blood, sweat, and tears, they've manually created all these rules. This has several problems. First off, though the students are all very careful and try to check their work, there's always the potential for a human error. There's also a problem with different people working on different applications, and they can make the rules that don't quite fit what you want. But the biggest problem is that it's very slow. How slow? Well, I applied these rules to the database KEG. It's just a database of enzymatic chemistry. Most of them are based from reactions database. And I found that only 45% are described by our rules. That's particularly bad when you find out that 5 years earlier when we did this, we actually covered a larger percentage of the database, even though we've almost doubled the number of rules. And this is because the database is growing faster than we can manually keep up making more rules. So we need some faster way of making these rules. So I was wondering, can we do this computationally? And of course, the answer is yes. Otherwise, this would be a weird talk. So the simplest way you can make, the simplest rules you can make simply consist of the atoms whose bonds were broken across the reaction. You have to have those atoms, otherwise it doesn't make sense. You can't break a carbon-oxygen bond if you don't have carbon and oxygen present. Fortunately for us, based on our system, these rules are actually pretty easy to obtain. We have our matrices, and all we have to do to identify atoms that break bonds across the reaction is just to look at our matrix and find any non-zero entries. So doing this, we can create a set of rules. And so for this example, this would be the rule that's created. I did this for the entire Metacyclic database. And not surprisingly, I found that I covered every reaction. So that's kind of trivial that I made these rules to cover every reaction that covers every reaction. What's more interesting is seeing which rules cover which reactions. So this is actually a plot of the Metastic database. Each dot represents 10 reactions. Each color represents one of these rules. And as you can see, there are these big groups of reactions that are all covered by the same rule. What you find is you actually have a diminishing returns. With just 5 rules, you can cover 14% of the reactions in this database. To get 50%, you only need 50 rules, but to get 100%, you need over 2,000 rules. This kind of supports our methodology behind these rules. There are some chemistries out there that are so common that they cover a lot of the database. But there's a problem with this approach. Look at, and this is a good example of what the problem is. So we're creating these rules just on the bonds that break. But in this example, if you just look at which atoms have bonds break across the reaction, you're just looking at these 2 carbons. So you're saying that you can always break a carbon bond. But really, this carbonyl and this benzene might be very important for this reaction to take place. Is there some way we can include this in our rules? Well, yes, but the problem is, what exactly do we include? Like I said before, different applications require different specificity. So for some applications like biochemical catalysts, you might want very general rules because you don't care if it actually works. But for other applications, such as if you're looking at the activity of a particular enzyme, you might want very specific rules. How do we account for this? Well, one way to allow this varying specificity without throwing in random parameters is to let the user group reactions that they think should belong together. So for example, these 3 reactions, there's several ways you can group them, and if we create an algorithm that, based on a group of reactions, will just generate the lowest cost rule that will describe all the reactions in the group. Here, cost is just the number of atoms that must be generalized. So if you just group each one individually, you cover all 3 individually. If you group these 2 together, you get a little more general rule. If you group all 3 together, you get an even more general rule. So I'm running out of time, so I'll go quickly. But there's a couple simplifying assumptions. Basically, you assume that the atoms that change bonds are the same across all the reactions, and you assume you only compare atoms that are the same distance from the bond breakage. This allows you to do a dynamic programming algorithm that's pretty fast, and I've implemented this to make these operators. The next challenge was, how do we decide on these groupings? Well, one obvious possibility was looking at genetics. As most of you know, DNA codes for RNA, which ultimately codes for these enzymes. Maybe if we look for reactions which are coded for, enzymes which are coded for by similar DNA, we can find groups of reactions enzymatic reactions that belong together. So I did this. But what was unfortunate is the lack of genetic coverage means you really only have the potential of covering 50% of the chemistry in the database, so you lost a lot. But this is also an opportunity, which is saying maybe we should take the time to look at this other 50% of the chemistry and find genes that are associated with it. My ultimate goal is to use these genetically grouped reaction rules and apply them to E. coli to generate a list of probable reactions, compounds, and genetic properties that correspond with those predicted reactions. This will allow industrial biologists to just look through this list and identify promising promising compounds that are easy to generate from E. coli, and it'll give them some hints as to what enzymes they might want to look at to perform those reactions. Hopefully this will dramatically increase the rate of enzymatic chemical research. To summarize quickly, I hope I've conveyed that enzymatic chemistry is important, that you can use these generalized reaction rules to develop intelligent hypotheses for probable enzymatic chemistry, I've developed an algorithm to automatically generate these rules, and I've used genetic-based grouping to try to associate the predicted rules with genes' genetic attributes. I'd like to thank my advisor, Keith, who did all the experiments, my advisor, Argon, who really got me into the genetic side of it, the group, and the CSGF, who helped not just with funding, but also I couldn't have done this without the increased knowledge of computer science, and a lot came out of my practicum. This whole genetic part came out of talking to Chris. So that's it. I hope I'm not too far over time, and thanks for your attention.